Hello, my name is Yusuf Sayer, and today I'll be presenting Chapter 1.5 Origin of Cells, IB Biology. To start off, we also need to remember that the cell theory it states three things. All living things are composed of cells, as in no living cells don't have no cells. Every single living thing needs to have cells. It also states that the cell is the smallest unit of life, corresponding to the very first thing it stated. And then it also says cells only arise from pre-existing cells, meaning cells cannot arise from nothing. They need to arise from something. Next, cells can only be formed by division of pre-existing cells. This explains the third sentence of the cell theory. Cells can only be formed by division of pre-existing cells. Cells multiply through division, and then this division means mitosis, which results in genetically identical diploid daughter cells. It also regenerates by meiosis, which generates haploid gametes, which is sex cells. Evident from Pasteur's experiments that spontaneous generation X cells and organisms does not now occur on Earth. Pasteur disproves the prevailing theory of spontaneous generation, his Swan Neck Flask experiments. Populations need to be seeded by existing populations. Cells come only from pre-existing cells. This diagram shows the process of the experiment that he did. The broth is boiled. Broth still remains free of microorganisms. The Guffnecracy is removed, and microorganisms grow in the broth, as in stuff coming from the outside. That's why. Cells can only be formed by division of pre-existing cells. What evidence do we have other than Pasteur's experiments to support this theory? I will show you other stuff. All known examples of growth, be it tissue, an organism or a population, are all a result of cell division. Genetic code is a universal. Each of the 64 codons, a codon is basically a combination of like three DNA bases. It produces the same amino acid in translation regardless of any of the organism. It doesn't matter. The logical deduction is that all the cells have arisen as the result of cell division from a single common ancestor. 1.5.2. The first cells must have arisen from non-living material. If we accept that there were times in Earth when cells did not exist, it is an obvious point that the first cells must have arisen from non-living material, obviously. The only other possible explanation is that life, in the form of cells, was transported here from somewhere else in the universe. However, there are some key problems. Non-living synthesis of simple organic molecules, for example, sugars and amino acids. Assembly of these organic molecules into polymers, formation of polymers that can self-replicate, enabling inheritance, formation of membranes to package the organic molecules. Earth's atmosphere was reducing in the early days. It did not contain oxygen until plants started photosynthesizing. So the plants themselves, they created the oxygen. The atmosphere contained a few gases. The list is hydrogen, nitrogen, water vapor, methane, ammonia, and hydrogen sulfide. All these gases, they came from volcanic activity. These monomers, basically, we call it a primordial soup because they all mix together. So these monomers mixed in the primordial soup, shallow oceans laden with chemicals, where it is thought that they react to form biological molecules. Miller and Urey tried to recreate those conditions in the lab in 1958. They were trying to demonstrate chemical evolution and the formation of more complex molecules from simpler stock in the primordial soup. The primordial soup, it refers to the gases I mentioned before and whatnot. They combined in the molecules from the previous page in a closed glass vessel to like recreate the conditions from the before to see if life would be created. The diagram on the right shows how they did this experiment. They after they put it in a closed gas vessel, as they show on the right. They heated the water so that it represents the volcanic activity that used to occur in the early days of Earth, and sparked electricity through the gases, stimulated lightning. After a week, they found that 13 of the 20 naturally occurring amino acids had been created out of the primordial soup that they created. Around 15% of the carbon was now in organic compounds, like the atmosphere. 15% of it has been used to create organic compounds, such as amino acids. They show you those compounds on the left. Non-living synthesis of simple organic molecules. Um, Miller and Duryea, they recreated the conditions of pre-biotic Earth in a closed system. These conditions included a reducing atmosphere, which means low oxygen, uh, high radiation levels, high temperatures, and electric storms. The heat, it represents the lava. The electricity, it represents the lightning, and the low oxygen, it represents the reducing atmosphere. Water was boiled to form vapor and then was mixed with methane, ammonia, and hydrogen. The mixture of gases was exposed to an electrical discharge sparks to simulate lightning. The mixture was then allowed to cool, and after 1 week was found to contain some simple amino acids and complex oily hydrocarbons. Based on these findings, it was concluded that under the hypothesized conditions of very biotic Earth, organic molecules could be formed, but no life is formed. 1.5.3 The origin of eukaryotic cells can be explained by endosymbiotic theory. The endosymbiotic theory, it explains the existence of several organelles of eukaryotes. This theory, it states that the organelles, for example, mitochondria and chloroplasts, originated as symbiosis between separate single-celled organisms. There are videos that are linked. I will link them in the description of the video. Development of the nucleus. Prokaryote grows in size, develops folds in its membrane to maintain the efficient surface area to volume ratio, which you will learn about in another video. The unfoldings are pinched off and forming an internal membrane. The nuclear region is enclosed in the internal membrane and hence becomes the nucleus. This is shown in the diagram on the right. The diagram on the right, it progresses as is shown. An endosymbiont is a cell which lives inside another cell with mutual benefit, which is like the aerobic bacterium, which is absorbed by the endosymbiosis. The development of chloroplasts would be a very similar process, except the benefit to the cell would be glucose slash starch instead of ATP. Be down below. They mention the development of mitochondria, and the subsets. An aerobic bacterium enters a larger anaerobic prokaryote, possibly as a prey or a parasite. A parasite is something that needs a host to survive. It survives digestion to become a valuable endosymbiote. The definition is on the right, as I just mentioned before. The aerobic protobacterium provides a rich source of ATP to its hosts, enabling it to outcompete other anaerobic prokaryotes. As the host cells grow and divide, so does the aerobic protobacterium. Therefore, subsequent generations automatically contain aerobic protobacterium. The aerobic protobacterium evolves and is assimilated to become a mitochondrion. Thank you for listening to this. I hope you found it useful, and I will see you in another video.